So I think one of the most effective ways of minimizing oxidation is just to vacuum pack the meat and educate the consumer that this is natural color.
Welcome to MeatsPad, a platform to share breakthrough knowledge that is accessible to the entire meat industry. Be ready to hear from meat specialists who will talk about numerous topics in meat science, including animal welfare, meat production, meat quality, and so much more.
This podcast is brought to you by the Niche Meat Processor Assistance Network,
the U.S. Meat Export Federation,
the National Provisioner, UltraSource, the new standard for innovation, and Dry Age Pro makes dry aging in-house, flexible, safe, and affordable.
Hello, meat folks. Welcome to the MeatsPad podcast again this week. Very excited because we have again a very, very well-known meat scientist, internationally known, and we're going to start more conversations with meat scientists around the globe as well. It is my pleasure to introduce Dr. Aladin Bekhit from the University of Otago. Welcome, Dr. Aladin. How are you today?
I'm fine, thank you. It's a great pleasure to be hosted by you, Francisco.
It is my pleasure, Dr. Bekhit. Really, I got to meet you during the ICON/RMC this year, and we kept that communication going, and we're here. We're really happy to have you on. Tell us, please, a little bit about yourself, a little bit about your career, your background. So how you end up being in New Zealand, so the audience can get to know you a little bit better, if you will.
Okay, so I finished my first degree in Alexandria University in Egypt, and my major at the time was engineering. So I'm engineered by training, and then I developed keen interest in food. So I moved on to the UK to do my master's in food process engineering at the University of Reading. And then I started to become really very interested in biochemistry of muscle food. So I moved to New Zealand, where I did my PhD in meat biochemistry. And I was very interested in meat discoloration and any biochemical natural systems that can support color stability for longer time. So I did my PhD at Lincoln University in Christchurch, New Zealand.
Great. Awesome. Thank you. Thank you for sharing that. We have a very interesting conversation with you today. We have very broad questions on some of your work as professor and meat scientist. But we'll start off by just going from the known to the unknown. Talk a little bit about what is oxidation, and you mentioned a lot of your work has been on meat discoloration. We can touch on either the protein or the lipid oxidation, depending on how you want to do it.
Okay, so that's a very interesting question, and let's start by saying that oxidation is basically our enemy when we start talking about meat quality or food quality in general, because oxidation can cause degradation of the nutritional value. So we eat food because we enjoy it, as well as it delivers certain nutritional value. So losing the nutritional value is undesirable. Also, oxidation will lead to severe changes in flavor and how the food is perceived and enjoyed. Also, this might be also leading to formation. Oxidation might lead to formation of harmful compounds that might be also causing some health issues. So overall, oxidation either is happening to carbohydrates, lipids, or proteins, will eventually lead to negative aspects. Very, very rare occasion where mild oxidation might be leading to production of certain flavors that can be desirable, like aging cheese, or in some cases, salamis making, or dried meat product, where a bit of oxidation and can lead to desirable flavor notes that normally consumer like. But generally speaking, oxidation has harmful effects on nutrition, health aspects of food, as well as the sensory attribute.
Great. So I guess on a couple episodes ago, we talked a little bit about myoglobin and color, and an introduction to meat color with Dr. Ramnathan from Oklahoma State University. And we we touched on some of the fundamentals of meat color, but I guess we can go a little bit more in depth about what that is and what is important for the meat industry. What are some of the strategies that we can use as a meat industry to prevent oxidation?
Okay, so basically oxidation happens in meat when the meat is exposed mostly to air. So that will trigger a series of oxidations that might be causing oxidation of lipids or other proteins. And there is very good evidence that we have been known for a long time that there is reciprocal oxidation between lipids and proteins. So if one is oxidized, it will cause other component to be oxidized. And this will be a continuous, what they call it, ping pong reactions. And this have been known for a long time. So color is very important quality attribute, as you mentioned before. And it's probably a very good early indicator of oxidation, because once oxidation or browning is occurring, this is a good sign that antioxidant capacity of the meat is start to become declined, and it will be giving us some hints about the onset of oxidative reactions. So in terms of oxidation and color, exposure to oxygen is one of the mechanisms, but there's so many other mechanisms like, exposure to UV, high temperature, and so on. And we find that really vacuum packaging can really take care of everything. So if you vacuum package meat and you store it for a long time at low temperature, chilled, that will be really taking care of any excess exposure to oxygen. So oxidation will be minimized. So from my point of view, I think just vacuum packaging the meat can minimize oxidative process to a great deal. Now, consumers mostly are not familiar with the purple red color of vacuum packed meat. And that is where the problem is arising, is that they take the bright red color as an indicator of freshness, and that is desirable by consumers. So I think one of the most effective ways of minimizing oxidation is just to vacuum pack the meat and educate the consumers that this is natural color. However, there's so many different strategies that have been proven in the past. So increasing the antioxidant capacity of the muscle through feed. The most successful example here is vitamin E, feeding for certain periods. So it's about 120 days for beef. This has been really useful. In terms of processed meat products, there is a wide range of natural antioxidants can be obtained from plants material. They can support delayed onset of oxidative processes. There is new wide range of active and smart packaging systems that can support this line of delayed onset of oxidative process.
I think we were talking earlier about, and I think you brought up excellent points about oxidation and meat color. And then you mentioned that you're up in New Zealand. Here in the US, when you go to a grocery store, you may encounter various types of packaging. You may find also vacuum packaged meat, and a more like a whole muscle, like brisket, or you look in pork, like a pork loin, a Boston butt in a vacuum package. We also have the famous PVC film, but it's aerobic packaging. In terms of what you guys do in New Zealand to prevent discoloration and oxidation, it might be different than we do here in the US.
So aerobic packaging still the most dominant one, because again, consumers, when they go and buy meat, they expect the bright red color. However, slowly over the last 7-8 years, introducing novel packaging systems, especially that contain vacuum packaging, has been a good way to add value. So normally loins with new packaging systems that include vacuum packaging will be probably about 40-60% more expensive than the traditional tray and the PVC aerobic packaging. So still the mainstream aerobic, but more and more meat companies find a niche in having vacuum packaged within a cardboard package that communicate a useful message. And this is regarded like upmarket or, a niche part. It's exactly the same meat, just the packaging system and the protection that is provided by having a vacuum packaged meat is just adding more value, and it will give longer shelf life. It will provide much better protection, as well as communicating clear messages about the product itself.
Absolutely. So you're mentioning vacuum packaging as being one of the becoming more popular in terms of that message that you just mentioned. So, and by looking at vacuum packaging, just looking at the steaks, so you see more of the steaks being vacuum packaged and looking at that purple color.
Yes, so more and more we try to educate consumers. This is the natural color of the meat, and there is nothing wrong with it. So previously people would think that is fault during processing, or it's not fresh, but more and more consumers start to accept color of myoglobin, the oxymyoglobin, as the normal and natural. And consumers start to become more aware about the safety regarding aerobically displayed meat and potential contamination, or even just allowing aerobic bacteria to grow. It's less likely to happen in vacuum pack, where most likely good bacteria, mostly if the product is processed carefully and under strict hygienic conditions, it's very likely vacuum packed will not have much of pathogenic microorganisms that can thrive under aerobic conditions.
We'll be right back.
The Niche Meat Processor Assistance Network, or NPAN, is a proud sponsor of the MeatsPad podcast.
Great. I think we're going into the middle of this, and we may just take a little break and ask you, Dr. Bekhit, tell us a little bit about experience that you encountered that you went through during your career. Again, it could be related to meat science, or just a personal experience that really had an impact on the way you look at things professionally and emotionally.
So as I mentioned earlier, my background is engineering. So I did engineering degree at Alexandria University. When I started my PhD, which was on the biochemistry of meat color and finding ways to stabilize the color, I had to learn lots of biochemistry. And one of the greatest things that I start to appreciate in life is that when you are really keen on learning, it's tremendous way actually to pick up knowledge much easier. So my perception and intake of biochemistry, which was really heavy subject for me at the start, but because you are keen to learn, it was massive learning experience. It's the intake was much greater than just sitting in a class forced to learn and then go through exams. So it was a great opportunity. However, the engineering side helped me to a great deal to look at things from a different angle. So when I was doing my early experiment looking at metmyoglobin reducing system in lamb, I was very keen on start looking at modeling and kinetics and this issue which I was comfortable with, as well as trying to look at rationale from engineering plus biochemistry point of view. So that was really good. So I think the experience being with different background supported my learning in the field of meat science greatly.
Absolutely. Thank you a lot for sharing that. And I think a lot of people can relate to that. I guess going back to again, the packaging, and I forgot to mention that another aspect of this trend that you guys are seeing over in New Zealand about going towards the vacuum packaging. You have a lot of benefits, more shelf life, but also we have a sustainable aspect, right? Consumer education, and you communicate that purple color, it's a natural color. And one thing along those lines that I want to ask you some of your work has been looking at the impact of oxidation, looking at the impact of oxidation on the microbiota. Will you please tell us a little bit more about that, to actually like relate for consumers that maybe they start to looking more about vacuum package, because as you mentioned earlier, we don't see a lot of oxidation occurring in vacuum package meat.
Okay, so in terms of research I did myself, I did on different proteins, like we are interested in developing novel proteins, and we look at the impact of these proteins on gut microbiota and the else, but specifically for oxidative processes occurring in meat and the subsequent impact on gut microbiota, we haven't done much, but there's some clues available in the literature that can give us some hints. So if protein oxidation is occurring, and that will make the protein is harder to digest in the stomach, and much of this oxidized protein become available for digestion in the intestine. This will have a greater impact on the gut microbiota, especially if there is no carbohydrates, which is it is preferred source of energy in the intestine. So if the gut microbiota is obligated to thrive on oxidized protein, that will start to generate some unwanted compounds. So one of these compounds that we just recently finished a review article on it for a journal. It's basically total volatile basic nitrogen, and that will have tremendous impact on cardiovascular diseases as well as brain health. So for me, when I look at discolored meat, a steak, it's probably related to desirability in terms of it looks nice or it doesn't look nice. It might tell me how long it's been sitting on the shelves, but I'm interested in what will happen after I cook and consume this product. And as meat scientists, we normally focus on meat quality and the quality issues during processing, during retail, and we don't tap too much on that part. Just very recently, some research start to be looking at different types of proteins and included oxidized meat proteins, mostly myofibrillar proteins, oxidized, and start to investigate the impact on gut microbiota. So very, very recently, but I think this is very interesting area that we need to keep investigating, because this will probably complete the cycle of our understanding about oxidation in meat products. So we understand oxidation in terms of flavor, in terms of perception of the color, some related to digestibility, but we don't really focus too much the downside of the whole process about bioavailability of nutrient and impact on gut microbiota, because more and more we start to realize actually gut microbiota is very central for the whole health and well-being of humans and animals.
Absolutely. I think this is a great topic, especially what you just mentioned. I mean, we know a lot of research, and not only on meat, but a lot of research on looking at gut microbiota and the impact on health. I'm looking forward to more research on this to, as you said, finish the cycle. When you look at meat quality, but also looking at how these oxidative compounds will also affect the gut microbiota and will ultimately have an impact on your health. Thank you for sharing that. First of all, I want to congratulate you, because you are mentoring 10 PhD students. So I think that's awesome. I don't think you mentioned it on.
This is in the last year. This is in the last year. So.
Last year, that's amazing. I think that's going to put a lot of things into context, so our audience can understand how busy you are, and we appreciate that you given us the time to talk to us today, and so we can provide some of the answers to meat processors, but because that's our audience, small and meat-sized meat processors that might have some questions. But I have one that I think you've done or conducted some research on blood waste at a processing facility. And I ask you that because last week I received a call from a meat processor in Texas asking about some of those procedures that we have in place, because when we call it maybe the gray water, when you have after a harvest day, you have manure, you have blood, you have water, you have all types of products in the water, and they were having some issues and concerns about how can they dispose of that water. But I think when I was reading some of your work, I think you may have a lot of good input on this. You start looking at, again, more sustainability and other strategies to take advantage of blood. Please tell us a little bit about that, and maybe we can follow up on some of those answers that you have.
So my interest in blood, basically, it came from the fact that blood is already consumed around the world. So if you go to Asian countries, they have blood tofu, which is coagulated blood that they can slice similar to tofu, and they consume that. So it's a product there, and they've been consuming that for hundreds of years, thousands of years. In Europe, a blood pudding and different recipes where blood is one of the ingredients. It's well known, and it has been again consumed and used for a really long time. It just our ability to consume blood is limited by two things. One is religious belief. So for Jewish and Muslims, they are not allowed to consume any blood, and that's clear in their textbooks, as well as consumer acceptability. So more and more we started to lose touch, new generation, about acceptability of meat co-products, and blood is one of those. So the best way to get all the nutrients that is available in blood is to invent ways of transforming the blood. So rather than being the thick coagulated mass or the red liquids that we can see, why we don't transform it into something that is more acceptable? The analogy is being used nowadays by the insect protein industry. So insects not appealing, transform it into a powder, a flour, and now you have something that has no resemblance with the original material. And if you don't tell someone came from that, they will not probably be, too much concerned about it. So I was interested to look at bioactive compounds from blood, because in the East, especially in countries like Korea, Japan, they believe, they have tradition of consuming blood with toasting wine and in celebrations and stuff like that. And they do really believe that their blood hold better health benefit compared with others. And I was very keen to examine blood from deer, pigs, sheep, and cow and see similarities and differences upon hydrolysis and the release of peptides and free amino acids. Is there any differences? What type of bioactive compounds can be extracted by utilizing either gut or non-gut proteases? So it was very interesting project, and I was hoping that will stimulate more research, which is happening. So we can see more activities about harvesting blood, separation of blood component, hydrolysis of blood, and some of this research can be quite important because, as you know, blood will contain high amount of ferritin and iron, which can be playing very important health role, not for everyone, but I can see young women and pregnant women as one of the targets for that.
Absolutely. And you brought up iron. What other compounds are readily available in blood?
Yeah, so iron, protein content, digestibility of protein will generate several important bioactive compounds and peptides that is not normally, the sequence is not available from meat itself, or from plant sources, so it has some unique peptides, and this peptide might be playing very important role in carrying something like iron, make it more bioavailable. So there's peptides. There is also lipids that can be available, although we don't try to include this in our research because we are mostly focusing on protein part of the blood.
And you mentioned hydrolysis. What is hydrolysis? And you mentioned peptides, and we know proteins are built out of peptides. I mean, would you just very broadly tell us about that?
So hydrolysis is the breakdown of proteinous material or breakdown of compounds. So in this case, we are interested in proteins that is available in the blood. And we have two different types of protein, either coming from the plasma or from the red cells itself. So the envelope of the red cell contains protein, and they are different. So the plasma composition and the protein sequences are completely different from the proteins that we can get from the red cells. Now, we are able to break down and degrade these proteins into different peptides that have different sequences. And many of these peptides can exhibit either one or more than biological functionality. So many of these peptides will be acting as antioxidant, so that is useful. It can also control blood pressure. It has even more advanced therapeutical effects if certain peptide was targeted and separated and investigated properly.
Good. And when you develop this project, do you guys look at what do you have to have in place in your plan to collect blood? What's the volume to be? I mean, is it really worth doing if I'm just harvesting 20 head a week or 20 head a day? So do you guys look at some of that?
So we didn't look at very much about the economics and viability of harvesting the blood, because it's a big issue. So if you are producing, high volumes, it's difficult to harvest and deal with all these volumes, and it will require some sort of investment. If you are doing too little, again, it's probably not worth it. I think here in New Zealand, because we are really focusing mainly on meat and some co-product, but blood is not one of these. And as you mentioned before, blood go to the green water, which is basically will be mixed with so many things, and normally it end up being used as fertilizer. So what our work was trying to add value by recognizing that blood will have unique proteins that might be upon hydrolysis generate something that is useful, and we might be able to capture some value from that stream. So definitely harvesting the blood and separating it and treating it in hygienic way is probably one of the biggest problems under the current production systems, because the plant is basically set up in a way that it will treat blood as a waste or a co-product of low value. And this lack of hygienic treatment and handling is a big challenge, as you mentioned.
We now know there's other people out there doing research and looking at how can we better utilize blood. You brought up some really good things about antioxidants and some other compounds that can be utilized maybe in the pharmaceutical industry or other types of industry that may be interested in that. Well, I think we're coming to the end of this episode. I want to thank you, Dr. Bekhit, for being here in this podcast again. It's amazing how, you're over in New Zealand, we're here in the US, and we can communicate, getting together with other meat scientists and getting to know what other people are doing, not only in the US, but also around the world. I think that's very valuable. And we thank you again for being on this podcast.
It's all my pleasure, and thank you very much for having me. I do really enjoy your effort in terms of, trying to have collective global meat scientists together. And I think it's a wonderful initiative, and we hope more people, just do similar effort. Thank you.
That we end this episode. Thank you a lot for listening. If you'd like to receive notifications on the new releases,
and the new episodes, please subscribe at www.meatspad.com. If you're a small and mid-sized meat processor and you have concerns or questions about a certain topic related to meat science and meat processing, please email us at info@meatspad.com. Thank you, and see you the next time.